Okay, on this slide, we're going to take a look at the 5 stages of fetal lung development. First, let's take a look at the picture. This is a picture of what's called an embryonic disc, and this is where the embryo has implanted into the uterine wall. You see the maternal blood pool there and the chorion. That eventually is going to become the placenta. Now, the placenta is a respiratory organ for the fetus. So you see the chorion there, it looks like blood vessels. Well, that's exactly what it is. They're eventually going to become chorionic villi, and that's going to be where gas exchange occurs within the placenta. The baby's blood and mom's blood never mix. So to the left of the picture, the embryonic disc, it's separated into 3 layers, the endoderm, mesoderm, and ectoderm. And in the boxes, it tells you which each layer is eventually going to develop into. So that will be important for you to review that. Okay, so let's take a look at the different stages of fetal lung development. The first one is the embryonic stage, and this occurs from conception until week 6. Some major things occurring here is actually the overall development of the airway. Obviously, you have the oral airway and nasal airway that's developing, the pharynx is developing. The lung starts as a small pouch, and then it eventually branches out into the right and left buds. By the end of this stage, you have 2 branches of the left lung and 3 branches of the right lung where the lobar bronchi have formed. During this time, the diaphragm is starting to develop. However, it is not complete until the pseudoglandular phase at week 7. Some other things that occur during the embryonic phase that are of interesting value here is esophageal atresia can occur during this time, and this is the result of an incomplete separation of the esophagus and trachea. And so around day 24, those two should separate. If they do not separate, then the result is esophageal atresia. If the diaphragm doesn't completely form and close the way it should, this can result in what's called diaphragmatic hernia. So if this abnormality occurs, it's going to be occurring during this stage. The pseudoglandular phase is our next one, and that occurs during weeks 7 to 16. Again, the diaphragm completely develops by week 7. Some other major things that occur during this time is the vocal cords appear and develop. We have major lung growth. We have cilia developing in the airway, as well as some goblet cells and bronchial glands. The cartilage at this point is still immature. By week 16, 25 generations of the bronchial tree should be complete. Interesting to note, around week 13, the chemoreceptors in the brain are responding to PO2 and PCO2 levels. And what this means is as mom's PO2 and PCO2 changes, this stimulates the fetus's chemoreceptors, and their diaphragm is going to start moving to push the fetal lung fluid in and out of the body, just like it was breathing if it were outside the womb. And so it's starting to practice breathing. It's doing fetal breathing movement, although it's not actually causing gas exchange because the gas exchange is occurring in the placenta. The next stage is the canicular period, and this is weeks 17 to 26. Some of the major things that are occurring during this time is the terminal and respiratory bronchioles are multiplying, the airways are growing in length and diameter, and this is a huge time for vascularization right now. By week 20 to 21, we have capillaries present, and by 24 weeks, 23 weeks, the capillaries are actually close enough to the air sacs to allow for gas exchange. Around weeks 22-23, we have the type 1 cells and type 2 cells present. At this time, immature surfactant is being produced. The chemical is PC, and that stands for phosphatidylcholine. That is just one of the components of surfactant. So although surfactant starts being produced at this time, it is immature. And let's go into the saccular period. That is weeks 27 to about 36. During this time, true alveoli are starting to appear. The lungs are very well developed at this point. During weeks 35, you have mature surfactant being produced, and this is the chemical PG, which stands for phosphatidylglycerol. And so once you have both PC and PG present at a ratio of 2 to 1, meaning 2 times as much PC as you have PG, that's when mature surfactant is being formed. The last period is the alveolar period, and this is from 36 weeks to term. And this is where the number of alveoli increase, and pulmonary surfactant is produced in increasing amounts. Let's go on to our next slide. As mentioned in the previous slide, the lungs for the fetus in utero are not used for gas exchange. It's the placenta that does the gas exchange. So what is in those lungs? Well, it's called fetal lung fluid. It's made up of protein and bicarb. It's higher in sodium and chloride, and it has a low pH. It comes from the interstitium of the pulmonary circulation's microcirculation. This happens because chloride ions pass into the epithelium, and the electrical potential difference causes the fluid to flow out of the cells. So it secretes about 250 to 300 milliliters of fluid per day from this interstitium, and this is always occurring. As the lung tissue develops, the secretion occurs. The function of this fluid is to maintain the patency of the airways and keep them compliant while they're developing. Without the fluid, the airways would fuse together and you wouldn't have airways that would work. They would be closed. The other function for this is it helps establish the functional residual capacity, with about 20 to 30 milliliters per kilogram of fluid being in the lungs at term. It's important to remember that the lungs secrete and produce its own fluid. And I talked about this on the previous slide, but the fetus is going to mimic breathing in utero, pushing the lung fluid out of the body by its mouth into the amniotic fluid. And this is why we can take the amniotic fluid to test for lung maturity, because the fetal lung fluid is constantly being sucks amniotic fluid in and then pushes the fetal lung fluid out as it mimics respirations. Now, during delivery, the fetal lung fluid is supposed to be evacuated out of the lungs as it's squeezed through the thorax. As it's squeezed through the birth canal, the thorax is compressed, and they believe that's how the fluid is expelled. However, about 1/3 of the fluid will remain in the fetus, and the lymphatic system just absorbs it over the next 24 to 48 hours, typically after birth. Let's go on to our next slide. Okay, now I want to talk about surfactant. Just to review quickly, fetal lung fluid comes from the pulmonary circulation. It's secreted by that, and it is released as the lung cells develop. Surfactant, on the other hand, is developed by type 2 cells. They secrete the surfactant, and then the lamellar bodies absorb it and resecrete it, so it replenishes itself and just recycles itself. It's made up of phospholipids, phosphatidylcholine and phosphatidylglycerol. I mentioned this before, but I'll just remind you, PC, or phosphatidylcholine, is present at around 24 weeks gestation, and it is considered immature surfactant. What that means is if the baby is born at 24 weeks with immature surfactant, then the surface tension inside the alveoli is going to be very high. So it's going to be very hard for that baby to take a breath and open those alveoli. This is going to cause an increase in work of breathing, an increased respiratory rate, and increased accessory muscle use, leading to respiratory failure. Now, PG, phosphatidylglycerol, that develops around week 35. At this time, it's considered mature surfactant. So if a baby is born at this point, their lung compliance should be normal, and it should be very easy for them to take a breath. They shouldn't develop respiratory failure at this point because the surfactant there is mature and reduces the surface tension. Now, surfactant production can be disrupted, meaning there are things that can slow down the production. So even though immature surfactant is produced at weeks 24 and mature surfactant is produced at week 35, if something causes hypoxia or hypothermia or acidosis to the mom or the fetus, then that fetus's ability to produce surfactant is going to be diminished, and so the surfactant may be delayed in its production. So what that means is just because a baby is born at 35, 36 weeks, we typically think their surfactant is mature and they're not going to have a problem breathing. But if they experience something during gestation that would have disrupted production of the surfactant, then even though the baby's 36 weeks gestation, if they're born and they develop respiratory failure, it could be because their surfactant wasn't mature because something disrupted it. Okay, let's go on to our next slide. Now, amniotic fluid, yep, this is our third type of fluid. This is secreted by the amniotic membrane, and it is dynamic, which means it's constantly being replenished and absorbed. This is partially due to that fetus mimicking its respirations because it's sucking the amniotic fluid into the lungs and then it's expelling it again, mixing with the fetal lung fluid and surfactant. The composition of amniotic fluid includes albumin, creatinine, lecithin, sphingomyelin, urea, enzymes, and proteins. The function of the amniotic fluid is to protect the fetus. It protects it from trauma. It also helps thermoregulate it so it's not exposed to extreme temperatures, and it also facilitates movement so the fetus can move around. If there isn't enough amniotic fluid, we call this oligohydramnios, and this can cause the limbs to actually fuse together. It also can cause a condition called hypoplastic lung, and that's where parts of the lung fuse together. Clubbing of the hands and the feet are possible. If you have too much amniotic fluid, this is called polyhydramnios, and this is where there's more than is needed. This is a predisposing risk factor for something called cord prolapse, and that's where the umbilical cord actually passes through the birth canal before the baby does. It's also associated with esophageal atresia, and they're not for sure why that is, but they just know they tend to go hand in hand. Let's go on to our next slide. Okay, let's take a look at neurological development and brain development. The brain is one of the earliest to begin developing. It starts development around week 7, but it's one of the last to be completely developed. It's completely developed after birth. At week 9, the neurological development, it can cause fetal movements. At week 10, it starts doing its first practice breath. By week 13, the chemoreceptors are activated, which means the fetus is going to mimic breathing in response to changes in PO2 and PCO2. By week 25, heart rates are going to accelerate with stimuli, which is an indication of the neurological control. By weeks 20 to 27, the fetus responds to sound. By week 36, there's fine auditory recognition. The last to develop is the auditory and visual. Visual doesn't develop until after birth, actually. Okay, I want to talk about a couple of the structures inside the brain there. First, the germinal matrix. This is a weakly supported and highly vascularized area at the surface of the lateral ventricles. Its function is to assist in the rapid cellular formations. It begins to disappear at week 34, and by term, it's completely gone. Structurally, it's not very stable because it is prone to hypoxic ischemic injury, meaning it can be damaged due to lack of oxygen, and the blood vessels are very prone to rupturing. They're not very stable. To add to this, premature infants are not able to autoregulate their cerebral blood flow. They haven't developed the autoregulation yet, which means any increase in systemic blood flow is going to increase their cerebral blood flow, which can cause that germinal matrix to rupture. For example, if you have a baby born at, let's say, 25 weeks, and you have them on the vent and you go to suction them, that suctioning is going to increase their blood pressure. Well, their brain can't auto-regulate to adjust the blood flow to the brain, and so what can happen with that spike in blood pressure, it can actually cause one of the blood vessels in the brain to rupture. We call those brain bleeds or intraventricular hemorrhages. So we want to take some precautions with these kiddos since they cannot auto-regulate in their brain yet. High CO2 levels can also cause an increase in cerebral blood flow, so you never want to allow them to have high CO2. It's going to increase the risk of those blood vessels bursting because of the increase in blood flow. Okay, another structure I'd like to talk about is the cerebrum. It's the largest portion of the brain and it consists of two hemispheres. It controls all voluntary muscular activities and consciousness, learning, memory, reasoning, judgment, intelligence, and emotions. Within it, we have the gyri, which are folds located on the surface of the hemispheres, and then the sulci, which are furrows which separate the gyri. The purpose of the gyri and sulci is to increase surface area of the brain. The brain starts out very smooth, and then increases in the gyri and sulci start to form around 22 weeks gestation. The presence of these and the abundance of them indicate fetal viability, meaning the fetus is more likely to have a functioning brain that's working correctly. Let's go on to our next slide. Fetal circulation. This is amazing. Okay, there are what we call 3 shunts in fetal circulation, and we're going to identify those on this slide. So first I'd like you to take a look at your picture and let's just identify some of the structures here. You see the placenta there to your left, and then you see the umbilical cord coming off of the placenta. Oxygenated blood leaves the placenta via the umbilical vein. Yes, vein. It is opposite of adults. Oxygenated blood enters the fetus through the umbilical vein. The umbilical vein, once it enters the fetus, it becomes the inferior vena cava. And right when it enters the fetus, it's right at the liver. Right here you see our first shunt, and it's labeled letter C on your picture. It's called the ductus venosus. And at this point, 50% of the blood entering the fetus, 50% goes to the liver, the other 50% continues on up the inferior vena cava to the heart. Now, the 50% that went to the liver, once it goes through the liver, it joins back to the inferior vena cava, and it eventually goes to the heart as well. Then we are at the right atria. So where the inferior vena cava, where the blood comes into the right atria, this is where our second shunt is, and it is labeled letter A in the picture. That is called the foramen ovale, and what that is it's a one-way flap. So blood that enters the right atria, it can flow through the foramen ovale into the left atria, and it's kind of a little flap that's hanging open. Now, the majority of blood actually goes through the foramen ovale. Because of the trajectory of blood flow, it's kind of the path of least resistance there, so the majority of blood actually goes through it. The rest of the blood that does not go through the foramen ovale, and most of this blood is what's returning from the brain through the superior vena cava, it's going to go straight into the right ventricle, then on into the pulmonary artery, and this is where our third shunt is located, and this is labeled letter F in the picture. In the pulmonary artery, the blood that enters, it can go through this shunt, and this shunt is called the ductus arteriosus, and it's connecting the pulmonary artery to the aorta. The majority of blood that enters the pulmonary artery goes through the ductus arteriosus into the aorta. Only 10% of the blood that enters the pulmonary artery perfuses the lungs and is returned to the left atria, then to the left ventricle, and out the aorta. The blood that enters the pulmonary artery and goes through the ductus arteriosus goes straight to the aorta and then back down and eventually to the umbilical arteries and returned back to the placenta. So one of your activities this week is going to be to detail fetal circulation from the placenta through the fetus and back to the placenta. And what you want to make sure you can do is identify where each of the shunts are located and where that route of blood flow is going to go. Okay, let's go on to our next slide. Okay, so we talked a little bit about the fetus having very low PaO2. But here I wanted to give you the actual values, and this is pretty amazing. In the fetus, the umbilical vein, remember that's where the oxygenated blood is, the good blood, the pH is 7.3 to 7.35. So it's a little on the acidic side. The CO2 is right around 38 to 42, which correlates with our venous blood. And then the PO2 is around 26 to 32. Now, to make this easy on myself, I remember CO2 around 40, PO2 around 30 in the umbilical vein. Now, the umbilical artery, notice your pH is 7.2 to 7.29, and your CO2 is 48 to 54, and your PO2 is down to 12 to 18. Again, to make this easy on myself, I remember CO2 around 50 and PO2 around 20. So, umbilical vein is the higher oxygen content with a PO2 around 30. Now, how can a fetus survive off a PO2 of 30? Well, for one, their blood cells are special. The red blood cells in a fetus have a higher affinity for oxygen than does mom's or any adult's red blood cells. And this will last for about the first 30 days of their life until those red blood cells die and disintegrate. But fetal blood cells have a higher affinity for oxygen than do mom's, and that's part of the reason why it can tolerate the low PaO2. Okay, let's go on to our next slide.